refer back to them. All right, so we are recording now. So tonight, sorry if I'm distracted, I'm still admitting people in. So we have 44 people so far tonight, so this is going to be a popular one. So tonight we are doing Morels and friends, all the creatures that are kind of closely related to Morels, or that we at least associate with Morels. This is a show and tell type of thing that we do, where we are going to share our screens and show off our finds, our favorite observations of Morels and other Morel-like organisms from years gone past. I don't think anybody's actually finding anything like that around here quite yet. So you can do that by sharing your screen, or if you've already emailed me, I have some emails from some people, and we will look at that. And I'm inviting everyone to, as you're sharing, to talk about how you found these. I know this is always a common thing that people really want to know about Morels, is how you find them, tips and tricks and stuff like that. So I invite anybody to share those types of things with everyone. I'm going to start out by, I'm going to share my screen. So I had a presentation I did last week, and I'm going to go through it really quickly, just as a really quick review, because we're talking about ASCOs here. Can you guys see my screen? If you can see it, give me a thumbs up, Dave, because I can see you. Okay, great. So this is a very quick primer from last week, or a review from last week. We are talking about Ascomycetes tonight. So Ascos are the mushrooms, well, the group of fungi is a really large group of fungi, and they reproduce, in a nutshell, by using these asci. These are sacs where the spores fill the sacs, and then the spores are ejected out of them, either out of the end, through like a little opening door. Sometimes they just rupture and come out and the thing just explodes. There's a whole bunch of different ways that happens. The way that these are different from a lot of the things that we call mushrooms, a lot of them that we call the Basidiomycetes, is that those mushrooms are producing spores on the end of these basidium, or basidia. Excuse me if I get my plurals and singulars mixed up here. I never keep that straight. But the spores on these guild fungi and these other ones that we call Basidiomycetes are produced on these structures called basidium. Whereas, as I was showing in here, the spores are produced inside these sacs called asci. So that's the main difference. And all the things that we're talking about tonight, Morels and Gyromitra and Helvelas and all of those things, fall into this group, the Ascomycetes, or Ascomycota. And we were looking at this tree that Jacob Polk created. He's a guy on Facebook who's a very good taxonomist, fungal taxonomist. He created this tree. He pointed out that it's not perfect. It doesn't have everything in there, but we were briefly talking about this last week. And last week we were jumping around with looking at Ascos from all these different groups. But tonight, I think we're probably predominantly looking at stuff in this Basidiomycetes. This is a class, and that contains, I think, everything that we're going to look at tonight. Morchellas, Gyromitras, all that stuff. I'm pretty sure Helvela's in there, although don't quote me on that. But I'm pretty sure it is. And then the only two resources that I really pulled out for tonight to help identify these things is the Ascomycetes fungi of North America. This is, I think, the gold standard for identifying Ascos in North America, by Michael Bugh and Alan and Arlene Bassett. A really good book, reasonably priced, full of color photographs and keys. And then this paper here, there's a lot of Asco literature out there. A lot of it's really technical. You can really get deep into the Ascos. They're heavily studied. But this paper here, this is a pretty good paper that came out in 2012, and I think it's still fairly accurate. Michael Kuo, who a lot of people know from Mushroom Expert, he put this out with some other people, not just him. And it was a revision of the Morchella in Canada and the United States, where he created some of the names that I believe are actually used in this book. So you can see the link here, and it's open access. So if you copy this link, screenshot this, you can follow this link. And you can find this paper to download and look at. And it has some photographs in there and some good descriptions. All right, so I'm going to stop sharing. And again, this is a show and tell. So we're just showing off. We're gonna get started here. What we generally do is we get a cue, So people just line up. So by typing their name in the chat. So if people want to share their screens tonight, start typing your names in the chat right now, and I'll start a running list, and we will look at that. And then at some point, I'll get into the ones that people emailed me. But is there anybody that wants to go for, oh, I'm sorry. Finally, if somebody could volunteer to type the names of the mushrooms that we're talking about into the chat, it helps everyone to see what we're talking about as well as just hearing it. So with all that being said, who would like to go first? Do we have anybody who wants to share their screen? Okay, I'm not seeing anybody jump in there. So the invitation will stay open. Please, if you want to share any morels or similar mushrooms that you've have found in the past, please type your name into the chat. I'm going to share my screen.
How to share the screen?
I'm sorry?
How to share?
Oh, okay. So to share the screen, at the bottom of your screen, when you move your mouse around, you move your cursor around, there's a toolbar at the bottom, and there's a green button. At least on my computer, it's a green button right in the center, and it says share screen. You have to have your, whatever you want to share, you should have open already. So have your photographs open, and when you click that share screen, you'll see your photographs there, and you just click on them.
I put some pictures.
So are you going to share now?
Yeah, I try.
Okay, great.
I don't know where it is. No, something wrong. Go ahead. You go ahead.
Okay, I will open up mine. Give me 1 second. I think all right. Can everyone see my email, or do I have to stop again?
I just see a list of ascos, boletes, chanterelles, trumpets.
Oh, good. I think you're seeing it. So Dave, I think you were the first one to email me. So do you want to go first?
Didn't look like emails though.
I'm sorry. Yeah, I need to unmute. Yeah,
Okay.
yeah. If you want me to go first, I'll go first. I have a lot of stuff. We don't need to go through all of it, but
yeah. Well, let's pick like 5 or 6 really good ones, and then if we come back around,
go from the top. Then I think I kind of prioritized from the top. Oh, right. I have to show this. This was the earliest that I have ever found any morels here in Northeast PA. March 23rd, 2012. It's hard to believe given the kind of weather we're having this March, but that was very, very warm March. And these, 1 or 2 of these were kind of old. Look at the one in the lower left. That one's old. That one's been out for a few days. That morel came out during winter. I'm pretty sure. And very unusual. These are black morels, Morchella and Justiceps. Older field guides will call them Morchella elata. It's a European species. Morchella conica. I'm not even sure that's a valid species anymore at all, but it might be a European species. But most of the Eastern North American black morels are Morchella and Justiceps. They don't start out black though. They start out very pale, kind of fleshy colored. They darken as they mature. The ribs darken and the pits remain kind of this pale brown color. So these are all Marcella and Justiceps. And I have seen these in New Jersey. Tulip poplar woods, mixed woods. Sometimes who knows where you'll find them, but they're not as common as the small yellow morels in New Jersey. Because I have picked morels in New Jersey over the years, so I know a little bit about where they're found. Also, occasionally you will find black morels in spread wood chips in ornamental sort of gardens. Those might be a different species. And it's actually likely that they are, that the mycelium probably came along with the wood chips. These are forest species, mainly tulip poplar, white ash, if there's still any white ash around where you live. We used to find a lot of these in mixed tulip poplar white ash. Once all the white ash died, almost all of our white ash in my morel spot here in Northeast PA has been killed by the emerald ash borer. And in fact, while those trees were dying, there were a lot of black morels coming out in those areas where those ash trees were. Someone on Facebook posed a question not long ago about, since the ash trees are dying, should there be a lot of black morels? Well, here in Northeast PA, that's past tense. The trees are dead, and we don't find as many of these black morels as we used to, say maybe around 2008, the decade before 2010. So these are a little smaller than the big yellow morels. They're a little bigger than the small yellow morels. They're
And-
all good to eat. They
How's
should
it-
be cooked.
I'll
Don't
say-
eat morels raw.
Don't you usually find these? Maybe you said that. I didn't catch that. Usually you find these are the first ones that you almost always find, right?
These are the first ones. Yep. These are the first ones. Some of the older field guides will say half free morels come out first, but I have never found half free morels previous to finding black morels. These
Oh,
come
I
out
always
first.
used to. Always years ago, but that's a lot of years ago.
I'm sorry, what was that, Susan?
I used to always look for and find the semi-libras, the half-free morels, about a week before you found anything that was considered a real morel.
Oh, really? Even the black morels?
Years and years. That would have been 80s into the mid-90s.
That's what I find down here too in central New Jersey. Always get those semi-libras first.
Before black morels?
Yep. I don't find a lot of black morels. I find occasionally under red cedar, I find them, but I don't find a lot of those. I think I'm pretty far south.
Red cedar, huh?
They like that.
Oh, interesting. Things vary regionally to a great extent when it comes to genus Morchella.
All right. Which is the next one you want to look at, Dave?
Just go down from the top. This next one is a false morel. This is our most common false morel, Gyromitra corfii. Older field guides are going to call this Gyromitra fastigiata, which used to be, I think, like in the OK Miller field guide, Gyromitra fastigiata was really this and Gyromitra bruneo lumped together. And there's no Gyromitra fastigiata in North America, as far as is my understanding. Now it's a European species. These are really difficult to distinguish from Gyromitra corfii. But one of the best pictures I have here, one of the reasons why I wanted to show this one is, if you section a Gyromitra mushroom, you'll see here that the interior is made up of chambers. It's not one hollow tunnel sort of thing through the whole mushroom. It's individual hollow chambers that are walled off here and there. And back to the taxonomy, Gyromitra corfii and Gyromitra brunea are very similar. And sometimes there are morphological differences. Gyrometra brunea usually forms like a sharp pointed roof sort of cap. And Gyrometra corfii, which is this one, usually has a more sort of brain-like, non-distinct sort of cap. But sometimes they're hard to tell apart. But you can do it if you scope the spores. The spores on Gyrometra corfii, and I'm not sure if I have a picture of the spores for this guy.
No.
I'm not sure. There might be another corfii post there from Mushroom Observer, but I do think I included a picture of some spores someplace. The spores for Gyrometra corfii, at each of the poles or the apices, have a knob, like a one distinct knob. Gyrometra brunea, which I think that's coming up maybe next, looks an awful lot like this. The spores are approximately the same size. They're shaped very similarly. I think the widths might be a little bit different. So this is corfii. But the Gyrometra brunea spores have ornamentation not only on the poles, it's sort of spread out. It's these little sort of lumps that are attached to the spores that are most notable on the poles, but sort of phase out toward the midsections.
Excuse me, Dave.
And you only see the ornamentation on Gyrometra brunea spores if you mount in H2O. If you mount in KOH, the ornamentation dissolves. I found that on Mushroom Expert, actually, Michael Kuo's website. So that's corfii.
I think you have a question, Dave.
Yes, I'm sorry, go ahead.
Thanks. Dave, when you say the ornamentation, you mean morphology or the shape there, or what, on the outside, or what?
Stuff attached to the perimeters of the spores, like the outer portions of the spore. Is there a picture for this one? There might be another corfii that I put on here, Luke, where the spores are, and this one really looks quite a bit like brunea. Oh, so is there another corfii?
No.
Yeah, there's a second corfii. This second, I show the spores there. It says spores. This locally, yeah, that one. Yeah, so we're gonna, now this, you can mount these corfii spores in just about anything. This one's kind of darker too. This one you might confuse possibly with esculenta. But another thing about Gyrometra corfii, a lot of times it doesn't have a well-defined stalk. Okay, so if you blow this one up, Luke, if you just go to the little square yeah, there we go. Move it over. Do you see on the poles here of these spores, like on each of the thinner ends? You see there's these little one knob on each pole? That's corfii. That's Gyrometra corfii. Gyrometra esculenta doesn't have that either. But these single sort of knobs, one on each pole, is that's Gyrometra corfii. I think corfii spores are probably a little thinner too, not quite as wide as but I forget. So I'm going to review now because we could look at brunea next. I think I have brunea coming up next.
Dave, what are
Corfii,
those?
supposedly you can eat it. I wouldn't eat it. You know what? Honestly, I ate corfii once. I dried it out. I rehydrated it in boiling water and I fried it twice. It was ridiculous what and it was like, oh, okay, so it was pretty good,
I think for any of these things that we call false morels, if anybody's interested in eating them, you should do a lot of research first.
Yes.
Don't
Now,
eat
brunea
anything
has a
like
bad
this.
reputation. Do you see how the brunea cap has like these sharp edges? That's the main morphological difference. And a lot of times brunea will have like one distinctive depression right at the top of the cap, sort of like this one on the lower left. But otherwise, this looks an awful lot like corfii. I've seen Brunia in New Jersey. These were found in Center County, Pennsylvania. There's a spot I go for morels, and these all come out during morel season. These ones that I've included, they all come out during morel season. And I find these Brunia every year in the same spot in Center County. It's a spot with a lot of elms. Now the ornamentation is a little harder to see here. So if you look on the poles, you'll see little nondescript sort of bumps on the spores, and those bumps will spread out a little bit more than you would see on the Corfii spores. So if you have a little bit better microscope, I have a better microscope than this one, but unfortunately I don't get good pictures through it because it's binocular. But you will see if you get a good view of Brunia spores, you'll see lumps that sort of, they're most prominent on the poles, but they spread out a little bit more. And you saw the Corfii spores a little before. There's just one distinct knob on each
Dave?
pole.
Dave,
Yes.
could you please describe the habitat for the
Corfii
Nigritas?
I found in a lot of different places, hardwoods.
Okay.
I found Corfii also though in conifers. I found Corfii in conifers up in Vermont one year. But I usually find Corfii like in oak woods. But it'll grow any place. I found it in apple orchards.
Thank you.
Brunia hardwood. I always find in hardwood spots with like elms and oak. You know, places I look for morels. You know, that's where I find these things.
Okay.
So,
Thanks.
how widespread is, you know, how diverse is the habitat? That's hard to say. I'm not even sure these are mycorrhizal. They might be some sort of saprobe. I don't know.
All right, well, before we close the gyrometers, I just wanted to point out, if everyone looks at this again, I know Dave said it on the first one, but see the interiors of them? So these are the
Yeah.
ones that people generally are calling false morels. And one of the quickest ways, if you're not recognizing it by this stuff, is when you cut them in half, these things have all of these pits on the inside, these chambers on the inside.
Yeah,
Whereas,
now there's another kind of false morel that I'll cover as well, the varpa. There's some places around here get a lot of varpas. But first, I guess I have an actual morel here is next. Or if you want to show the varpa, show the
Yeah,
varpa conica.
yeah, look at the fur.
Yeah, show that one. I find these in near old apple trees. It's the same habitat as a lot of yellow morels. Now they don't really look that much like morels. A little bit. Sometimes the caps are a little more pitted. These are Verpa conica. Notice the interior is almost one hollow chamber. It's not quite hollow though. It's got this pithy stuff inside. Now that pithy stuff disappears as the stalk expands. That pithy stuff will dissipate, and when these get a little older, they will look like they're hollow. Now notice how the cap is attached to the stalk only at the extreme apex of the cap. So you have to see the attachment there. Now look here, this is a little bit of an older one. This stalk appears to be hollow. There's only a little bit of pithy stuff left in it. Now there's another species, Verpa bohemica. I don't find it around here. They get it in the Midwest and they get it out west. That looks more like a morel. And in fact, I've had discussions on Mushroom Observer with people in California where we're trying to decide, is it Verpa bohemica or is it a half-free morel? Sometimes it's hard to tell, but at least our Verpa conica it doesn't look as much like a half-free morel as Verpa bohemica. So I've got a few other things too, as much as you want to show us. Oh, here's a half-free morel, Eastern half-free morel. Got one right up there. So this is Morchella punctipes. It used to be Morchella semilibera, but that's a European species, so we had to come up with a name for our North American half-free morel. And there's a different name for the one out west of the Rockies. You can see this has the more like regularly, the pits and ridges like a morel should have. It's called punctipes. The species name is punctipes because you see on the stalk there's all these little like sort of warts or grainy sort of things. That's punctate. So that's what's referred to as a punctate surface. It's got these little raised sort of dots, grainy sort of things. So that's why it's Morchella punctipes. And I think they're good edible. Some people say they're not that good, but I dry them out. And I put them in soups and stuffings and things like that, and I think they're good.
And you find these in hardwoods, like where you would find regular
Hardwoods.
morels.
You know what? These are really weird. I find them in hardwoods, but one year, this was a long time ago, maybe 1998 or 96 or something. I used to go to New Jersey to pick morels, and
Poaching.
friend of mine and I were at a place in New Jersey, in North Central Jersey. We found 400 of these in like a 1 acre area, half acre area under hemlock trees. So go figure. These might be a saprobe. They might be a saprobic species. So they just sort of pop up different kind of areas, usually in hardwoods, usually in the spots where I find other morels, but that might be because those are the spots I go to, when morels are out.
Yeah, good point. All right, you want to look at one more, then we'll move on to somebody else for a bit?
Yeah, you know what? Show this Morchella diminutiva. This is probably the most common morel to be found. You know what? Somebody else probably has those. Yeah, that's good. Let's move on. Let somebody else go.
Well,
Okay.
one question, Dave. You mentioned the first time you found, the earliest you found the half morels was March 23rd.
No, that was black morels.
Oh, it was black morels. Okay. How late have you found them?
The latest I ever found morels that were in decent condition was like May 28th, but
Wow.
in a cold area in Northeast PA. Like
Thank you.
in a colder than average area, in a colder than average spring. One year I found a whole bunch of morels around a dead elm. It was right around Memorial Day, I guess. But they were mostly too far gone. It was a tree that I had driven past once or twice, and I didn't think to stop. I was going trout fishing and I didn't stop. And finally I said, okay, I'm going to stop and look. There were like 50 really big morels by this elm tree. I didn't even know it was an elm actually back then. And that was when I learned, like, check the elms, the dead elms.
All right, cool. Well, thank you, Dave.
Yeah.
Okay,
Any
so-
other questions? Okay, let's move on.
all right, so the next person that emailed me was this person, Yi Chao. I apologize if I'm not saying your name correctly. Yi Chao Ching.
You are right.
I'm saying it correctly?
Yes.
Oh, good. All right, so I have your photographs that you sent me here. So if you would just, I'll show them if you just want to narrate and tell us what we're looking at.
This is a Chinese farmer do the greenhouse.
So they are cultivating them in China.
Yes.
Yeah, that's a really cool picture.
Yeah, look at that.
Wow. And probably not gritty or dirty.
What's in the bags that you have that's scattered on the field?
Yeah, do you know what's going on here, Yichao?
This is like a natural bag.
That's pretty impressive.
This is after he cut, leaving the road only.
Those are cultivated? That's amazing.
After yeah, this is before.
Yeah, very cool. A farm. You know,
Yeah.
Tina Eller, who is a good friend of our clubs, she's a professional mycologist for a mushroom farm. She told me a couple of years ago that her company, Phillips, she said that they know how to cultivate morels, but they don't do it because it costs so much money to do it, and they're so lightweight they can't make a profit on it. So she said they decided not to do it here in Pennsylvania with their
What's
farm.
in those bags? Is that mulch or compost or?
I don't know. I don't think anybody knows. Well, very cool. Thank you, Chow, for sharing that.
Thank you.
Luke, Tina Eller gave me morels in October once, about 2005, when Phillips was still cultivating them. But what you said, it was not profitable for them to upmarket it. But they were nice to eat in October.
Yeah, I bet they were, huh? All right, Penny.
I'm sorry, were those cultivated morels you were just showing?
Yes, they were photographs from a farm that Yi Chao was showing us, a farm in China.
Oh, they I think the species is Morchella rubrobrunius or Rufrobrunius or something like that. They're a wood chip morel. They're a saprobe, so that's why they're able to grow them. But when you buy a kit to a morel growing kit, what my understanding is what they do is they put sclerotia into the substrate that you bury. The sclerotia are like the predecessor to the fruit bodies. So I just had to leave for a second. I just got back, so I just thought I thought I heard a question about that. I'm sorry.
All right, cool. All right, Penny, would you like to go next?
Sure. So I've only found this once, and I think it's a Helvella. And I wrote in my email which one I thought it was, but
Yeah, right. I don't know if you can see this. The file.
Macropus, but I
Yes.
don't know if that's correct. And I was reading about it, and they said that they were separating the ones with the short stipe and the long stipe to separate species, but then they combined them. But I don't know what the current thing is, and I hadn't gotten to this one. Was a couple years ago in the Adirondacks. There is a way to distinguish them because they have spindle-shaped spores, but I didn't examine the spores.
There are a few different species of these so-called stock cups that are all pretty much put in Helvella. There are other cups that have stalks, but not such prominently thin stalks like these Helvella. So I think there's Helvella elastica, I think is one of them. Helvella macropus, like you just said, and that's a pretty good guess at that one. There's a few other names too.
Yeah, I have the ASCO book out right now, and like the last line here to go along with what Penny was saying, it says the distinctive subfusiform, finely roughened spores easily separate Helvella macropus from other cupulate Helvella species such as Cupuliformis and Fibrosa. But yeah, they have a bunch of names in here. But this looks fairly close to what the Bissettes and Bug put under Macropus. Theirs look a little hairy. Not sure if we can see hairs on this or not.
We see this at least once a year in the Adirondacks, but down in New Jersey, we used to see it every once in a while years ago.
When do people usually find these? I don't find these until summer generally.
Yeah,
It
summer.
used to be
Late
pretty
spring.
wet, as I remember, to see these.
I think I remember finding these in the Poconos on one of our our Pocono forays in June, late June. All right, cool. Well, it's definitely a Helvella, right?
Yeah, I guess so.
Oh, it's Helvella. That's
Yeah.
Helvella.
I guess there's another picture underneath. Did you show that one?
Oh, yeah, I did.
Okay,
Here, I'll open
So-
it back up though, in case nobody saw it.
yeah. Okay, which one do you want to do next?
Let's go right in order.
This was found during morel season in the Adirondacks. I don't know what kind of gyromitra it is. Gyromitra means convoluted turban.
This is what's more common in the Adirondacks that I find very often at the same time that the rest of you might be looking for edible morels. This is much more common here in the Adirondacks. I call it Gyromitra esculenta, but I've never investigated the real literature.
Yeah, that's what I guess too.
It's been called Gyromitra esculenta for a long time, but there's been some recent research that suggests that might be split in North America. But this is what they've been called. The ones that really look like bumpy brain-like caps and stalk, usually a well differentiated stalk, and often under conifers. Susan, I'm guessing you get
Yeah.
these under conifers.
Yeah, that's where this was found. And then I just wanted to mention that gyri are what they call the surface of the brain, the convolutions on the surface of the brain. So,
Oh,
and
that's
this
where that
one kind of
name
looks
comes from.
like a brain to me, also. This picture.
It's a really nice looking one. They're often more convoluted than that. And that picture you had of the split open was quite interesting too.
Oh yeah.
Yeah, that's really cool.
Yeah, it looks like it had been eaten, and are these are not edible, correct, Luke and Sue?
Don't eat any gyromitras, regardless of what anybody tells you.
Yeah, I mean, I did read about this a little bit, and there are people who do eat them, and they go through all the various methods that Dave was talking about, but the toxin is very dangerous, and even if you cook them, there might be some in it, and there's evidence that people accumulate it in their body so that they may eat them for years
Right.
and then all of a sudden die
Yeah,
of it.
it's a cumulative poison. And I would have said
Mm.
monomethylhydrazine, same component in rocket fuel.
So there's a study going on right now by Alden Dirks. You might be familiar with his name because he did the lecture. Yeah, he did the program on crust fungi a couple of weeks ago for us. But he's doing a research project, and I really should have gathered the information up because he's actively collecting gyromitras to investigate the toxins in them. Because there's a lot of, it goes back and forth a lot.
Yeah, and they also say that the gyromitra in different parts of the world have different amounts of toxin.
Yeah,
Maybe-
I think that's true.
Yeah, and I think that's one of the things that he's looking at. So he's collecting from all over, at least all over North America, to study this stuff.
In Finland, you can buy a can of Gyromitra that's pretty much not much different than a can of corn you buy here in the United States. Just a
Kind of
metal
scary.
can with a label on it and a picture of the mushroom on the label. But the toxin apparently is highly volatile, so it cooks out really quickly. But as has been said, it might not all be removed by cooking. Also, there's an old folk tale a woman in Russia supposedly was boiling a bunch of Gyromitra esculenta mushrooms, in a pot. And she opened the pot and inhaled the fumes and died like an hour later. That's an old tale. It's a folk tale. I don't know. It's probably not well documented, but that's something that I have seen, in print.
All right.
I was in Finland, oh God, I don't know how many years ago, 20-something years ago, and we were at a fancy hotel and they had a wild mushroom soup on the menu, and we were there for 3 nights. And I ordered it every night. It was wonderful, tasted fabulous. And then when we got to our conference, which was way up on the Arctic Circle, and our mushroom friends there said, oh, that would have been Gyromitras. We've been trying to tell the Finns for years not to eat them. So I've eaten it and survived, but only 3 nights in a row in a row.
Wow.
Yeah, I wouldn't eat them if I knew what they were.
It's like the puffer fish that the
Yeah.
Japanese.
Marisol, is that contact for Alden Dirk's crust fungi or funegg.com?
I made a mistake. Fungi.
Okay, so
So
that's
sorry.
what I asked.
Thanks.
Yeah, so last year he was actively collecting Gyromitra, and I think he will be again this year.
That's
He's
what
been-
I'm saying. If I find them and he wants them, I can dry and send them and photograph them for him.
I think one of the problems with studying Gyromitra, in terms of the levels of toxins, is that they need fresh material
So he was
because
having,
of the volatility of the toxin.
yeah, he was actually having people freeze them and then send them. And he was suspecting that even, freezing and then thawing out a day or 2 in the mail would be fresher than drying them.
Hmm.
That was his theory.
So these are morels found with tulip poplars in eastern Pennsylvania, near where, not too far, an hour west from where I live. And I think these are the smaller yellow morels, diminutiva, diminutiva, and
You
and
found
they're
these?
supposed to be mycorrhizal as well as saprobes. Does anyone any comments if that's the correct?
Well, before you even said tulip morel, I could recognize it as a tulip morel because close to 40 years ago, this is what we found all over my part of Hunterdon County. And I used to find a lot of them. They always had that kind of thumb shape.
I see.
Tasted good, but they were only about 2 to 3 bites tall.
Yeah, they're not that big.
Rarely got very much taller than that.
And very thin stipes on them.
Well,
Does anybody know if the black morel or the yellow morel or the golden morel, which one tastes the best?
you're lucky if you find any morels or at all. So just to sort out don't worry about it. Just make sure it's hollow inside and you cook it really well.
I think
Yep,
there's
yep.
another
Different
picture
types
too,
seem
right?
to go better in different cooking strategies.
Mhm.
Like the big yellow ones, I will slice perpendicular to the height, sort of. So you get these like star-shaped wheel-looking things. They look really nice, and those are really, really meaty. So I would use those as like a side dish with meat or something. These little yellow ones, these diminutiva, and there's another species too. I think it's Scriptriformis. They're pretty similar to these, but they're usually a little
Scriptriformis, I read about that. They say the top is more egg-shaped.
yeah, the top
and
is
more-
supposedly, I think I find them in Maryland. I go to Maryland every year. I have a good spot down there. And I think I find both kinds, both of the species in the Maryland spot. And I have some posts on Mushroom Observer to show them. But these little guys, these little, the Scriptriformis diminutiva, I dry them out and I put them in like soups and stuffings and things like that. I don't cut them. I just dry them out whole. And they look nice. And of course they taste really good too. But
I just made a cream sauce and served with meat and with these, and I thought they were great.
yeah, they're all the morels are really good. The half-free morels, maybe used fresh, are not quite as good as the other types. So I would
Yeah, they're a
dry
little bit
all of
wimpy.
those. But
Wimpy
they're
flat.
all really good, and there's really so many different ideas you can come up with for
Living.
making morels.
Have you tried the black ones, Dave? Have you had the black before?
Yeah, yeah. Well, like I said, we have a local spot here in Northeast PA where we've only been finding maybe a handful every year for the past 10 years.
Mm,
But when the ash trees were dying and the Marcella organism, which was associated with those ash trees, was went into reproductive mode because that's what allegedly happens, and that seems to make sense to me. We were getting 200 to 400 black morels out of our local spot. And that went on for
wow,
about 5 years. Give or take
That's
maybe there was
incredible.
like a bad weather year where it got too cold or something. We didn't get
They
that
Wow.
like
many.
it cold. Morels
But
like it cold.
Yeah, I'll just say I've seen the same thing, where the ash trees are starting to die, where you have a couple of years of really good hunting and then all the trees are dead, unfortunately.
it looks like most of the black morels in my area were associated with the ash. Some of them are still hanging on in spots where there's really big tulip poplar trees. But yeah, oh yeah, I like the black morels. I dry them out. I would use the black morels fresh. Also, I make well, it's a recipe based on a recipe I got out of a Bissett book, Edible Mushrooms of Eastern North America, I think it's called. There's recipes in it. They have a recipe for quiche. So I've used that recipe and I put all sorts of things that I forage: morels, leeks, wild leeks, fiddleheads. We have asparagus out by our yard here, so it's not really foraging. It sort of grows, near our house. But I put all that stuff into a quiche, and it's really good.
With any morel, I feel you should cut it from top to bottom to make sure that there's nobody living inside. I also hold it up to the light to be able to see in the pits and ridges whether there's ants, slugs, other little critters, little tiny thrips, and brush them out with some kind of small brush. I dry them without washing them. Or
Yeah, I don't wash them either. I do. If it looks like the big ones, especially if you have a big one and you can't see inside it by shining light on the outside of it, sometimes you can see if there's any problems in there. And sometimes you can like bang the morel, the bottom of the stalk, and stuff will fall out. But yeah, I agree, Susan. Sometimes it's the best thing to do is just slice them vertically so that you can make sure there's nothing in there. Also, if you find a morel that's got a centipede in it,
oh, yeah.
it's probably not going to be any good. The centipedes emit some sort of like iodine smelling secretion, and it gets in the mushrooms and it'll ruin a whole meal. So
Are
you
they,
really have to watch out.
are they
The
centipedes or millipedes?
I'm sorry, millipedes. You're right,
Yeah,
Luke. Thank you.
it's the
Yeah, the
it's,
millipedes, the big, thick, like crusty ones.
yeah, it's the cyanide, right?
Is that what it is?
When you shake them, they smell kind of like an apple almondy smell.
Yeah, they smell like iodine to me.
Yeah, I think it's cyanide.
It ruins a whole meal. You have to make sure you don't use a morel that's got any of that stuff on it.
That's
And
interesting.
usually that, when that secretion from a millipede gets on a morel, it usually stains it sort of bright yellow. So smell them. You can smell it. It's easy to smell.
Was there,
the-
was there another picture in there, Luke?
Yeah. Wait a minute.
Oh, and then you had said to say
Uh,
what you do to get ready for mushroom hunting or morel hunting. And so that was in the one day, all the morels I found, which was very exciting because the first time I ever found morels. And one of the books I had read said, look at a lot of pictures of morels before you go hunting. This is, I guess, for beginners, so that you get the picture in your mind, because when you're looking for them, you keep getting fooled by pine cones and things. So if you look at a lot of pictures, then you can prepare yourself. And I think that worked for me.
Cool.
Yeah, also you can, there's a mail order you can, that sells look-alike rubber morels or some synthetic thing. And they're really morel looking. And you can take them out, scatter them around, and then try to find them to get some practice there.
Another
That's
thing to
interesting.
do when preparing to go morel hunting is to be aware of ticks. And to we use permethrin on our clothes, and you should tuck your long-legged pants into your high socks and wear light colored clothing so you can see ticks when they're on your clothing. People think snakes and bears and things like that are dangerous in the woods. Well, I guess they are to some extent, but not nearly as dangerous as ticks.
I used to pick 10 or 12 of them off of me about every hour when I used to hunt morels in New Jersey, again, 15, 20 years ago.
Yeah, me too, 15, 20 years ago in New Jersey. Yep. But when I started using the permethrin, I don't see that many of them anymore. And when I see one, a lot of times I'll actually see it fall right off.
Well, that's good.
Yeah, it's toxic though. Permethrin is toxic. You have to be careful how you use it.
Yeah, I have cats, so I would be hesitant. But where I am here, the ticks are not prevalent really here yet.
You don't have them up in the Adirondacks, I know. They don't have them
Well,
up in
not
Vermont yet
here.
either.
In the Champlain Valley, they do.
Yes, the lower elevation. Yeah, you're right. Okay. I mean, yeah, when I go to Vermont, I'm usually up in the mountains. But yeah, in the Champlain Valley. Yep.
So the
Yeah, and-
permethrin, do you spray that on your clothing?
Yeah, I
before
take it
you put
outside.
on?
I make sure there's not a lot of wind. I make sure I'm upwind of where I'm spraying. I put the clothes on hangers, close them up, hang up the hangers on a clothesline. Try to stay, 20 inches or so away. And I spray I try to get every square inch of clothing, socks, pants, and long sleeve shirts. And then you let it stay outside for a while and let them dry. And we have a cat. So we're careful. We have what we call a mud room. It's like a little passageway from our back door into our kitchen. It's, there's doors on each end. So we put the sprayed clothes, we hang them up high inside that room so they're not in the house, so the cat's not going to sniff them. But yeah, permethrin is dangerous to cats. Apparently, is what I've read. There's warnings on the bottles.
Is DEET got permethrin in it? Because I heard that DEET was very strong like that.
No,
There's no permethrin.
two different, the instructions in the CDC say use permethrin on clothing only and DEET only on skin. They also say with DEET, make sure you've got at least about 25% DEET in the what you buy, because sometimes if you just pick up a container of off, they sell formulations as low as 7%. And so you've got to look at the label there and make sure you got something around the 25% concentration. But
Okay,
DEET for
thank you.
skin, permethrin for clothes.
Okay, thank you.
Permethrin too, once you sprayed your clothes, I do the same thing that Dave does. You don't have to wash them every time. It sounds pretty gross, but I usually use them a few times. And I usually store them in. I fold them up, I shake them out. Sometimes I run them through the dryer a little bit just to shake anything out. And I store it in a plastic bag in my closet, kind of hidden away, because we have a cat too. But that seems to work for about a month, they say, but I usually give it about 2 weeks.
Yeah, 3 weeks I find. By the time the 3 or so weeks goes by and it's like getting into like the later part of May, the ticks start backing off usually around then anyway. The height of the ticks seems to be April through maybe the first week of May. At least that's what I found.
Oh,
around
not
here.
here. Not
No,
here.
they're out all
Summer, when the weather is really hot, oh my gosh, the ticks are horrible.
Yeah, they're
In,
pretty horrible.
in all of the woods or just in the Pine
Oh.
Barrens?
Oh,
You're
in the
right.
pine barrens is really bad, but I
Yeah.
get less in the other parts that don't have pine trees. Yeah.
Yeah,
Oh,
that's what I've heard. I've heard the Pine Barrens are really bad
Oh my
the
gosh,
whole
they're
year.
horrible, and chiggers too. Whew.
I've heard about the chiggers. You can't even
Oh,
see
boy.
them. Now, there's
Right.
different kind of ticks too. There's the wood ticks, which are these big brown ones. They call them dog ticks sometimes. They're a little easier to deal with. They're easier to see. They move. They don't. They're not as quick to bite. And I've never had a problem with a wood tick. But I find those in the summer. I find those around here in the summer, usually early summer up until about maybe the 1st of July. But the really bad ones, the little black ones, the deer ticks, I find most of those in April, early May, and then they come back in October.
yeah, but they're out all summer. You have to be on the lookout all the time for them.
I'm
Yeah,
always
I-
on. I always
just-
look, but
I
I
was just-
hardly see them in the summer.
I was gonna say, I guess the bottom line is for us mushroom hunters is to really be careful of the ticks. So,
Yeah, may I say something? One time I went to the woods in November and I thought there were no more ticks, and I was playing with wood, and I came back home and I didn't check my clothes. So I got a tick and it was on my leg for 4 hours, and it gave me like a ring. So I had to go to the doctor and blah, 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 do the blood thing to see if I got the disease in November, and
Yeah.
it was kind of cooler.
yeah, I think you'll get ticks on you all year round as long as the temperatures
Yep.
go above 40 or so.
Yeah, I say October. October in my part of Pennsylvania, which probably means November in like central New Jersey.
Yeah, I've had ticks on me year round here in Pennsylvania, like
I had a tick
Philadelphia.
myself yesterday.
Yeah, so anyway, I wanted to add a question about the Morchella scriptiformis, because I never heard of that before. You say you find them in Maryland, Dave?
I'm pretty sure. Yeah, I should have made a post. I have some Maryland Morchella observations on Mushroom Observer.
Huh, I'll have to look
Yeah,
them
and
up.
I think I have found that one. And yeah, the caps are shaped a little differently. They're a little bigger too. They tend to be a little bigger.
Are they
Yeah, and
growing
I think
under?
it's like the egg-shaped cap. I think was what Susan said before.
Are they
and
growing under
I'm
a
pretty
specific
sure
trait?
that's my understanding. I don't think I posted any of those. I you know, I sent you like, I don't know, like 8 links, and then I said like, okay, that's enough.
Are they growing under a specific tree?
I read about them and it said.
Go
like-
poplar
like-
ash.
Like
Okay.
Dave said, that they are slightly bigger than the diminutiva and usually found in the Great Plains area. So
I see.
they're going to be less common around here.
Right.
Yeah,
Okay,
I think
cool.
I found them in Maryland. Tulip poplar ash, same thing. Same thing
Okay.
as like where you would find them in New Jersey.
Yeah.
The diminutiva. And
Right.
they grow together. If I'm correct about my identification, then I have found them right along with diminutiva.
Okay, cool. All right, Susan, you want to tell us about what you
Yeah, I'll tell you what I
Okay.
call them anyway. The first one here is, I call it Gyromitra esculenta. It's a little darker than usual because I think it's a little older. It's been out there maybe a week to 10 days, and I actually have found that they can sit out there even after rain for a couple of weeks. I sort of don't always pick them because I don't need them for anything, and I'm not going to eat them, but they do make a nice photograph. And Yeah, they're very common here in the Adirondacks, much more so than the edible morel. Next one, Luke, is, okay, this is taken a few years ago, maybe 10 years ago, also here in the Adirondacks. And it is also, I call Gyromitra esculenta. You see how it's more of the reddish brown color? That's more common than the one before. And you can just about see in the one down at about 4 o'clock, the sort of chambered inside look to it. I don't always cut them in half to look at them, but they're an interesting mushroom. When I was in New Jersey for 30 years, I think I might have seen this once. But up here, I see at least a couple every year. And this is always May into the 1st of June. You can see these here anyway. Now the next one was taken in October. This is a Gyromitra infula. And there was somebody alerted me to this along the trail that she walks every night. And there was about 15 of them all over these logs, which I think were aspen, tulip, I'm not aspen, poplar aspen logs. And they're, well, it's another thing I don't see very often. I don't remember ever seeing this in New Jersey, but here, this was quite a fruiting of them. By only showing you the one picture here. But that again was October. It's not a thing you're going to see in the spring. It's folded over. It's not, I don't know how to describe it. The stem, and then it's folded over kind of, and then turned up on the ends kind of to make that funny shape.
They sometimes call this saddle-shaped morel.
Yeah, it is kind of a saddle
false morel
because of the way it straddles the stem. And this is also one, as well as the other one, if it's in the right stage of spores, you can blow across them, or even when you just pick them and they'll throw up a cloud of spores. Okay, so the next email has Helvella's, I think. Yeah, this Miracell, I think, showed this last week. Helvella acetabulum. The wrinkled stem is what gives it away in the gray coloring. It's really an interesting little fungus. I see it about once a year, maybe not quite that often, but it's very easily recognized. And often there's bunches of them at the same time. The spores forming on the inside of the cup there, and this sort of little funny fluted stem that sits up high with the deep veins to get it up high enough off the ground. And you can see there that it's all pine needles, but I think it's still a saprophytic kind of.
But where I find it, there are no conifers.
Well,
Yeah.
it'd be an opportunist thing for whatever organic matter it can work on.
Yeah, enrich.
I don't honestly know, to tell you the truth.
Yeah, but it was surprising to me because where I go, there are no pine trees.
Well, you can see the pine needles right there. It was right under a white pine tree. And there was quite a few of them. And there's another spot on this property that's also conifers. There's nothing else around there. And they've come up in the same place. And it's always here about 1st June, end of May. After the morels or the gyromitras have come up, then I start looking for this and a few other things. So it's sort of, and then I have kind of a lull like you do in New Jersey before the real mushroom season here starts after the 4th of July, unless it's been tons of rain, and then it will start in late June. And I forget, the next one is what? Oh, this I see maybe once every couple of years. Helvella elastica, this is another saddle one. They're very distinctive because they have that smooth kind of saddle look with the hollow stem that's always kind of white and this kind of funny flute, I don't know what you call it, V shape, and the brownish color. And often there's bunches of them there. Saprophytic, I think, on the ground, often along the path where I walk, which is.
Are they edible?
I wouldn't eat any, you'd really have to look up and see. There's not much to them, so I would not probably eat them, but then that's not my major thing, edibility. I don't worry about that so much. I'm more interested in being able to identify them. Now, some of you on this next picture might laugh, and also especially since you can see, oh no, this is, I call this Helvella lacunosa. Every once in a while, about every 3 or 4 years, I see a bunch of these. And gray, fluted, kind of hollow inside or chambered like the gyromitras. The ones down in the lower right there, I've cut in half at least once. Also a saddle shapey thing, but always an irregular cap. It's not a true saddle like the ones before. We used to see these down in New Jersey. There's also a white version of this. And this past year, I saw the white one up here, Havella crispa. This is one that Elsa Valinka, I think, has been working on to try and figure out if this is a species here complex or a European name and we're using the wrong name. But I think they're a really cool mushroom. When you see them, you can't miss them. I mean, they're dark against the habitat soil. And here again, it's along a trail in conifers. But they're pretty distinctive to recognize.
When do you find those?
This was midsummer, usually midsummer. For here, that's July into August. Again, very weather dependent, a lot of rain, I think. I think that's kind of the determining factor of in mid-July, there has to be a lot of rain. Okay, Luke's. Well, last one picture is kind of, you would not recognize that's me in 1997, but we used to find a lot of morels in New Jersey under elm trees. This was the end of April in 1997, and you can see it was a really good hole. I was with Arlene Bissett. It's a long story, but anyway, so it was probably Hunterdon or no, Warren or Sussex County, but it would have been entirely under dead elm trees. But we used to find collections like that a lot through the late 80s into the 90s. If you read some of those old newsletters, you'll see Greta Turchik used to find thousands and thousands of morels, and she used to kind of boast about it in the newsletter. And then some of them got wise to that and sort of, where is she? She lived in Franklin in Sussex County, and that's an old mining area where it was crisscrossed with a lot of old abandoned railroad tracks. And the elm tree, being a pioneer tree, had come up along the old railroad tracks. For the most part, I think they're pretty much gone in New Jersey, the elm trees. By the time I left, it was very few that I could find.
I've got one elm spot about 40 miles from my house here in Northeast PA, right? I can still, on a good year, I'll get close to this many in like a 2-acre area. It's just all elms.
One thing, you folks who are looking for morels, I used to pre-scout my areas for where the old abandoned apple orchards were or the dead elm trees. I used to go before I expected to see them. And you know, that's another thing. We haven't talked about morel indicators. What are the plants that come up at the same time or are about out at the same time? Like
May apple.
Yeah, May apple.
May apple.
Apples are kind of doing this and are like right here.
I think bloodroot and Dutchman's breeches and blue, Virginia
Yeah,
blue.
there's quite a few. You have to pay attention when the dandelions flush. Bloodroot, just when the bloodroot finishes, the flowers and petals all fall off. After the forsythia, and remember, we can talk as much as we want to about time of year, but you have to be more observant to the botanical calendar. That is sometimes those plant and apple blossoms, when the apple blossoms are out. So that may be earlier. This actually is quite early for New Jersey to find that many that year. Usually I used to find Mother's Day used to be the best time for morels in North Jersey years ago.
What are the little flowers that are behind you in that picture?
Um,
Are those,
periwinkle or, what's the other thing that's a ground cover? This is in my neighbor's.
Spring Beauty, maybe?
No, it's not spring beauties because this is a planted,
I find hepatica flowers during morel season in the woods.
I'm trying to think, the red bud, it's in the pea family. It's the pinky, shocking pink flowers all over the trunks of an ornamental tree. It's in the sweet pea family. I can't think of the name of it. But when that's out, that's when the morels are out in New Jersey. And you can start sort of in South Jersey and, the end of April, or if the weather's good. And then as the calendar goes along, you start to go further north to look for your morels, so that you end up in Sussex County by third week of May usually. But again, depends on the weather. Anyway, I used to, if I got them big enough, I would stuff them with ground turkey and a little bit of shallots and breadcrumbs and a little bit of curry, and then bake them. Oh, it was so good, because then I could freeze it in portion sizes. But that's if you get an awful lot. Otherwise, the good ones I would always dry, and I used to donate them to my coppagy. So that's all.
That's an awesome find with all those morels. I think I would be so proud of myself.
See, all of my morel pictures are before digital, so I couldn't find them. I couldn't find any good pictures of years ago of a lot of morels, but we used to find a lot of morels once we figured out Greta's secret.
One other biological indicator, I find that when wild leeks or ramps are
Yes,
in season,
Yes.
that's what I find morels too.
right. If you have a good place for that, and up here, that's the indicator. There's only one spot up here that I have found morels in any quantity, and it's a south-facing slope. That's the other thing, that the morels will come up on a south-facing slope because I think they like the extreme temperature and the sun, so warm in the day and then cold at night. And here they're under ash trees, and ramps are out at the same time. So it's a place that a lot of people in my area kind of know, and they kind of stumble across the mushrooms when they're out looking for the buckets full of ramps. And they do grow like that here, the ramps. Okay, sorry, Luke, that's it.
All right, great, thank you. Dorothy, are you here tonight? I haven't heard you yet. Oh, she said she might not be here. Well, we'll look at her pictures. She had it labeled as diminutivas, I thought, but well, that's a diminutiva. Is that?
Yeah, that one might be.
That's not though, is it?
No, I don't know what I would call that.
Isn't that
Probably
a black
Americana.
Yeah.
It's an old Americana, I would
Yeah.
say. It could be an old diminutiva. Diminutiva can get big.
Yeah, I have some pictures.
And when they blow out, when they get old, they blow out like that.
Yeah, I have some pictures of some big ones, big diminutivas.
Yeah, but there looks like there's too many pits on that one.
Yeah, that.
So Dave, that was not a Augusta Seps like you showed first?
I don't think so, no.
How can you tell the difference?
The color just doesn't look right. An Augusta Seps that's that old, the ridges would be really dark. Notice that the ridge, the lines on the ridge tops are kind of light. And Justice Seps would be really dark, especially when they get really old like that. The very tops of the ridges are black. Also, it looks like there's no, well, okay, I was going to say something else, but I'm going to withhold because it's not true when they get this old.
All of the morels have been kicked around for a lot of years with a lot of different names. And I think somebody in Europe started to really divide them up or do some DNA work. So I think we kind of have to relearn all of the names or find out what is actually growing here in the United States. So we can call it whatever we want, but if it's hollow in the side and it's got pits and ridges, it's an edible morel.
Yeah, well, there were some pretty good studies though about 10, 15 years ago.
Yeah, there was somebody out in Washington who did a study. I sent mushrooms to.
There was, well, in the eastern half, the eastern 2/3 of the country, There was the Quo study, and there was a competitor at the same time, the Clowitz study. Clowitz is a French mycologist,
Yeah,
and
that
he
might be
partnered
what
with
I'm thinking
Chris Matherly from Georgia. And they were like competing with Michael Quo at all. And some of the new names are Quo names. Some of the new names are Clowitz names. But there are still some types that are difficult to distinguish because there's like cryptic species. There's this thing, Morchella ulmaria, that looks like a Morchella americana. That's what used to be Morchella esculenta. But the old names are all pretty much out the window, except maybe Anjusticeps, which I believe used to be applied a long time ago, but now that's the eastern black morel. But these look like either diminutiva or they look like diminutiva, these ones here.
They
Yeah,
look like they grow
Very
under
nice.
tulip poplar.
Yeah, these for sure.
Yeah, the
Diminutiva.
tulip morel, they call it sometimes because it grows under tulip poplar. And other hardwoods. Almost always hardwoods.
All right, Liz, how about you?
Yeah, I have to say that we still find quite a few morels in New Jersey. I'm in central New Jersey, but Hunterdon County, Monmouth County. And I find I do very well in like the floodplains of stream beds. And it's usually ash and tulip poplar. This was found under a kind of a dying ash. And it was really odd.
Are those entelomas growing on it?
No, they're not. I think they're pseudosclerotia. When you take them off and slice them, it just looks like a mass of hyphae. It was very, very damp. It rained a lot. And this was on the bottom facing the ground. You know, maybe little spores that had germinated and started to send hyphae out. But I had never seen anything like this. And we really get a fair number of morels. So I don't know, you commented on, I had it on Mushroom Observer, Dave, and you thought it might be a diminutiva, but the
Looks like diminutiva to me. I don't know what these little things are. I know there's these
Yeah.
like these parasitic entelomas that
No,
look like, but I guess that's not that.
yeah, I think it was like either a pseudosclerotia or maybe just spores that had germinated. I did go back and plant it. I thought, ooh, maybe.
I think your
Did
idea
you
of sclerotia is correct.
Okay,
If the thing was like this when you picked it.
that's exactly what it was like.
Yeah.
This is in my backyard. I didn't take a picture out in the woods, but I sent a picture to Tom Volk because he
Yeah, I was
talked.
going to say, he's the person that would have probably to.
That was just kind of an interesting one, but yeah, keep looking. They are out there. They don't like the pines. The soil's too acidic, although one of my friends did find one by an old blast furnace, and I guess the mortar that it deteriorated might have been alkaline enough. But they like calciferous soil. And I find them on the edge of the coastal plain where there's a lot of fossilized, like old oyster shells and stuff.
Yeah, Bob Peabody used to say that he thought it was always on the limestone ridges in North Jersey.
Yeah, Alamucci. Although I've looked in Alamucci, I've never found one there, but maybe everybody else got them all. Bob Hosh has a really great article on our website. There's a little section for stories, and it talks about one of his mushroom hunts with Bob Peabody.
Yeah, I saw that. Yeah.
Those epic days when the elms were dying and people used to get shopping bags full of great big ones, but it's still fun. It's like an Easter egg hunt. It really is great fun.
So I wanted to, Molly was asking if there's any groups that go out and hunt for morels in New Jersey. So typically this club in JMA does do a morel hunt every year. Unfortunately, this year we've postponed that because of the COVID stuff. We decided not to do that because it draws such big crowds. But typically, yes, we do go out. I don't know of any other groups in New Jersey that does.
Nobody wants to share their
the Institute Woods in Princeton is where we usually do that foray, and there's no reason you couldn't just go out on your own.
And the reason we go there is because even with a large group, usually a few people can find a few. It's the only real public place that you can count on at least a few morels and a lot of people.
Yeah, I guess a year or two ago we went and we got like 108 morels, and some of them, most of them were the diminutive.
On the weather.
Yeah, but some of them were pretty good size.
we found like 300.
Yeah.
I know because I asked a lot of people and I was adding numbers. But like some people found 100, some person found 100 alone, and everybody else like 110, 110, 5. Yeah, but only few people found bunches of them.
That's just the way it goes.
You
They
have
all
to
put
train
together.
your eyes for the difference in texture and color. And you know, it's a real hunt. You have to acclimate yourself to the texture of the morel if you're going to find them, be successful.
Okay, so I have a few here to share. This is the Americana, because we haven't really seen that one, Morchella americana. Or and I had to hunt around to find some pictures of them, because I usually don't see, for some reason, I don't photograph them. But these are the ones I find the most of, I think, here in the Philadelphia area. At least they're the easiest ones to find because they tend to get big. So you can see that's on, you know, sitting in the palm of my hand. My hand's fairly big.
What's the habitat?
That is a deciduous woods. So if you look at the ground, you can see that there's red garlic mustard in there. That looks like it might be a maple leaf, but it could be viburnum too. But always in like the tulip poplar type forest. Lots of tulip poplar forest around here in southeastern Pennsylvania. So that's where I almost always find them. I used to find a lot of them with ash trees, but there's not much in the way of ash left around here. But if you can find ash, the big old ash, like last year, I was finding some big old ashes right in my area, like in part of the parks that I've never gone to, and they were dying, but they were still growing there with them. Yeah, through this photograph, I very rarely ever find these Augusta steps. Is it Augusta steps or Angusta steps? There's an N in there, isn't there?
Yeah, it's N-A-N.
Yeah, Angusta steps. So these I found, again, in the tulip poplar forest. You can see all the lesser celandine, blankets of celandine, where I usually don't go bothering to look for them because I can't, it seems like that stuff mats so hard on the ground that I feel like I can't see underneath of them. But see, these were poking right through. I guess it wasn't quite as thick right there. But again, tulip poplars, that's the dominant tree there. Tulip poplars with like beech and oak. And then finally the diminutivas, which I find a lot of them too. But look at the size of these things. These things were 10 inches tall. I'm sorry, not 10 inches, 10 centimeters, not 10 inches, 10 centimeters tall. And I felt pretty confident these were diminutivas. So,
Yeah,
I'd say probably.
Yeah.
I mean, the scriptiformis, you know, I don't know how to tell the difference really. But yeah, diminutiva can get big under the right conditions.
now this is like a really nice spot that I find these in. It's along a creek at the bottom of like a nice gentle little valley. So everything kind of in the surrounding forest, everything kind of drains down into this creek, but it's not swamp. It's just kind of like a little bit of a lower, very fertile area that's south facing. So this whole valley gets like some nice warm sunshine, and they're this big giant tulip poplars, 200-year-old poplars. I know they're that age because I've seen ones that have fallen and been cut. I've counted the rings on them, so they're over 200 years old. And you know, the way I find them is I just, you know, I look for those big poplar trees, and I go up there and I get down on my hands and knees, squat. I do. I squat down to the ground and I start scanning from, you know, from the trunk outward, maybe 10, 15 feet until I find one. And I go to that spot, and when I collect that one, and then I look in a complete circle around me, just scanning the ground. And invariably, I'll find another one that I didn't see before.
And that's
I
called the Sam Ristich crouch.
The Sam Ristich crouch. Okay.
Yeah, he
Yeah,
used
and
to,
you have to be careful. If you see one, don't rush toward it because you might trample one or two.
that's right.
Yeah. Yep.
You have to stand still and scan around, and like
Yep.
you get in your hands and knees sometimes. I always used to take a stick because the best places were about up to your knee or higher with poison ivy. If the poison ivy gets up to your rear end, it's past the season for finding them in New Jersey.
All right.
Another thing you mentioned, Luke, that hasn't really been highlighted is areas that are low-lying but well-drained. Yeah, I don't really find them like where it's swampy, but near the edge of a swampy area where there's trees and where it's well-drained.
Yeah,
Yeah,
nice, like nice loamy soil where, where the creeks aren't really flooding up into, but getting close to it. So they
Right.
stay
So
nice
in
and
other
wet.
words, if the water, if like it's flooded in the spring and there's vernal ponds and then they go away, that is not a good area.
No, probably not. Usually,
Right.
like I said, the south facing slope, there was a theory some years ago that one of the things that actually triggered the morels to fruit was the leaching of nutrients after the snow from the soil. So the soil was kind of washed clean underground, and that was what helped to trigger the morels, and when it's somewhat cool too.
Someone asked earlier about if there were still any elms in New Jersey, and the only live elms I've found are we're in a very swampy area, too wet for morels to grow. And it reminded me that there's another botanical indicator, sort of the opposite. I find that if you start seeing skunk cabbage, then it's too wet to find morels.
Mhm.
Yeah, that's
So
true.
so when you get into that
I
area, you
agree.
want to kind of find some like the hills around there and just try to get couple feet up in elevation.
There
Yeah,
you go,
look for
yes.
little humps, mounds of, drier areas. And sometimes those areas are good.
Mhm.
Luke, before you go too far, can you help point out the difference between the Americana and the Diminutiva, please?
Sure. So, okay, so that's the Americana. They typically get larger. See how large that is in my hand, and they're fatter all the way around. And then there's differences. I may need help here because I would have to look in a book. There's a differences in the way the pits are in these. I think the Americanas, aren't they a little more scattered? See how they're
Not as many pits,
Okay,
not
so
as many pits and more elongated.
yeah, see how long they are in there?
Well, but in the Diminutiva, they're more elongated. This one actually, this one's a tricky one here, but it's an Americana. I agree. It's got that big thick stalk, the thick stalk base. That's a more of an Americana thing. Americana usually has a higher number of pits.
So let me go back to the other ones.
Yeah, go back to a Diminutiva. So the pits are really vertically sort of arranged, and they're really elongated.
The ridges are lined up vertically.
Yeah,
Yeah, that's kind of what I was getting at. See how yeah, just what Sue said. The ridges are much more vertically aligned. But yeah, I can see what you're saying too, Dave. These, it looks like you have these big pits, and then within the bigger pits.
yeah. There's not as many pits, and they tend to be a little bigger. Now, if they're very young, though, and the mushroom is smaller, their pits are going to be tighter. So it might be a little harder to make that kind of distinction. But when they're young, they're really small too. That's the other thing.
So does that answer the question? Does that help?
Absolutely, thank you.
Okay, you're welcome. Okay, so I'm gonna come out of here and I'm gonna stop sharing for just a minute and kind of make a last call for anybody who did not get to share. If you have something that you want to share.
I have a couple.
Okay, go ahead.
And Luke, I don't have anything to share, but I do have a question, some follow-up questions about apple orchards.
Okay, sounds good. So you want to go first, Dave, and then Lauren can go?
Sure. Am I on?
Yep. Okay, so those are a good example of what we were just talking about with the pits.
Yeah, this is under poplars. It's in a poplar grove. All of these are in poplar grove. There's some areas of the grove that are wet. I know Dave W was just saying avoid the swampy areas, but I found them in dried up swampy areas where there was maybe 2-3 inches of water, and as the water drained in the kind of a broken root of area. It was kind of maybe 2 feet around where the water was held. It dried up, and about 7 or 8 of these popped up. That was about 6-7 years ago. I did notice all my pictures over the years, all around the end of April. So
Looks
3
like a
years in a
tulip
row,
morel.
it was April 24th, 25th, and 26th. On those days is where I found them all. But I look in poplar groves, and that's I do well there.
What county are you in?
Camden, Gloucester,
Gloucester, down.
Gloucester, Camden. Those
Yeah, okay.
counties.
Mhm.
I don't really go north of Camden. I know Burlington. I've been a far way up there with you guys, but I stay south.
Yeah.
It's a very leafy area. There's some sweet gum in the area. There's some big old poplars. It's an old growth forest. The couple forests that I go in around when ramps are coming up, because I go down around Kennett Square, Pennsylvania, but not for mushrooms. I go down there for some ramps. There's a little secret spot there that we hit every year. So I get ramps first, and I come back up here and I get the morels.
What's a ramp?
A ramp is a wild leek. It's a very popular leafy green with a sort of bigger than a scallion bulb on the bottom. They're really just wild leeks. So I find my poplar groves, there's 3 or 4 in the area that I use, and very leafy. I always imagine when I find these, I put a stick in the ground, like Luke says, start looking around, and I usually find more, and I mark them all with a stick, and I go through the area. I spent a few hours in the woods, and I imagine if I had a leaf blower, I really feel like I'd find 3 or 4 times the amount of morels, because I find them peeping through leaves, and I can't move all the leaves out of the area to find what I'm looking for, but it's fun nonetheless.
Carry a stick and push the leaves out of the way.
Yes, that's what I use. I have a walking stick I use for my morels. If not, I'll find one in the woods and break it down to size. I know people feel that they're acidophobic. They like alkaline soil. Hence, I think that when there's a burn in some, morels, that the alkali from the ash makes them pop up, in my opinion. And some areas that there's some burnt areas in some of the woods I go into, but I haven't found any morels in that particular area of the woods. But I usually clear around it and get a picture of it before I. This is an interesting one. I'm not sure if this is a black or a blonde with black ridges.
It started to dry out.
Is that what that is?
Yes.
Felt very
Yeah,
fresh.
it's a blonde. It's a diminutiva.
Diminutiva. Okay. Yeah, so these are all various years over the last 2 or 3 years. My older pictures are somewhere else. Oh, that was last year. A small bag full of them. And then I'll take the bag. I usually harvest them in a net bag, a mesh bag. If it's a paper bag, I will take that bag and when I'm done with it, save it. When I go back out to the woods, I'll open it and shake it back out. Hoping some spores will make it out to make another morel.
Yeah, they look all to me like tulip morels.
Yeah, and again, they're tulip groves where I'm looking, and again, I clear the ground a little bit. That might be the same one. A couple of these look like reruns. There's one that's a little dried out. This was leaning against a log. And as I moved it away, it was kind of getting old there. A little dried out.
But still tastes good.
Still is good. A couple more. I'm trying to get through the end of this. Little bug got some of this one. Saved me a lot though. Saved me some.
Yeah,
Those
it looks
lids
to me
here.
like the
Getting a
slugs
little-
have gotten at those.
Little dry, a little old at these. A couple more. And that's my favorite dish. Crayfish morel. And that's all I have.
Nice.
So thanks for letting me share that.
Yeah, thanks for sharing, Dave. Okey-doke. So who wanted to ask about the apple orchards? I'm sorry, I forget who said that.
That's me.
Oh, hi, Lauren.
Hi. So I know we've talked about this a couple of times in the past, but bottom line, so I have a couple of orchards that I find morels in every year. And Dave, I heard you talk a little bit about testing soil. So my question is, if you go to an orchard and you find morels, do you eat them or do you not eat them? Do you only eat them if you can test the soil first? Like if you cannot test the soil, do you not eat them?
Well, I started finding morels in old apple orchards here in Northeast PA in 1994. Before that, I spent many years looking and not finding any, and I finally found them then. And I ate them. I ate all of them. I found out about this lead arsenate pesticide thing, I guess maybe about 12, 14 years ago. So I got some lead test kits that you can use with soil. Someplace in Canada I found online. It was not real expensive. I got like $35 for like 12 applications. And so I tested these small orchards where I hunt around here. They're not big commercial orchards. They're just usually a few acres that was just like adjunct to a dairy farm, probably just apples that the family gathered for themselves. So I think it's less likely that those places had been consistently treated with heavy doses of lead arsenate. But I got good results on my tests. Now, in the meantime, I have found a very large orchard not too far from where I live. It's one that I didn't know about until only about 5 or 6 years ago. And I didn't find many morels there until last year. Last year, I found quite a few one day. I found a couple of areas where the trees were dying. And I found some really nice big Morchella americana, the great big yellows. And I took them home and dried them, and I segregated them out. And I'm going to go back to that spot this year and get some soil and test it, because I haven't tested that area yet. But this big apple orchard I would be a little more suspicious of an area like this. It's very big. It's many acres of apple trees that are in rows. It was clearly, a commercial enterprise at one time. So I think it's probably more likely that an area like that had been consistently treated with this pesticide, which was legal and used extensively between, I think, like 1870 and 1965 or something like that. I'm not sure of that. But my idea is, and I don't have any research on this, okay? But the pesticide was lead arsenate, that we do know. So arsenic is very difficult to test for the presence of arsenic in soil. You have to send out to a lab. It's going to cost you at least $40 a shot. Lead is not that difficult to detect. So you can do it basically, you can detect lead levels, you know, just varying degrees of accuracy, I guess, but you're really just trying to find really bad contamination. That's the issue here.
And what
But
lead levels are considered acceptable and what lead levels
well,
are?
I forget what the concentrations are.
Okay.
But the kit will, you know, the kit, you know, you immerse some of the soil in water, but it's, you know, and add some of this chemical. And I can tell you this, that if it turns black, that's really bad. If it turns like orange, there's some lead present. You know, if it doesn't turn much at all, then it's pretty clean. Um, but my idea is that neither lead nor arsenic leaves an environment all that quickly. Those two substances hang around for a long time. So they're both dangerous. So you're either going to find both of them or neither of them. If there was contamination due to using this pesticide. So I figure that if I test for lead, that's sufficient to determine whether an area is safe or not. And like I said, the morels I found last year in this great big apple orchard, I have put those aside. I know which ones they are. I've labeled the jars where the dried ones are. And I still need to test the soil from that area. And so I'll make that a priority this year because I will go back to that spot this year.
Okay.
So to answer your question, I'm only eating ones that I feel safe eating at this point.
All right, does that help?
Yeah, yeah. Is that how everybody else feels too? Like, should I just not eat them if I can't test the soil first?
Unless you're eating, you know, bowls and bowls and bowls of morels from under apple trees.
No, just a few from, you know, each spot.
And you're, you know, that isn't the only kind of morel that you eat. I mean, I've eaten morels from under apple trees for 40 years, but not that many.
Yeah, no, I don't find too many.
If you're lactating or breastfeeding, do not.
Yeah, I don't
Honestly.
think that's going to happen to me soon.
Well, this is global. Sorry, Susan.
Well, we had somebody in the club, you know, who had heavy metal poisoning, and it was attributed to him eating morels from under apple orchards in mostly Warren and Hunterdon County about, it would have been late 60s, 70s into early 80s. And
he ate a lot, right? Like
yeah, he ate a lot.
And bags of them every year.
Exclusively from under apple orchards, because that's kind of where he lived in an area where there were a lot. And also he would get out the maps and look on the topographical maps from the 50s. You can see where the old orchards were. And that's where he, you know, that was a treasure hunt for him. And he ate lots. And he also gained a lot of weight. He gained a lot of weight, and he didn't have a problem until he started losing the weight. And apparently the heavy metals were in the fat cells. And when he started losing weight, that's when it sort of came out to affect the rest of his body.
One of the spots that I go to is believed that it might be an old, like, cherry tree orchard. I don't know how factual that is, but do we know anything about, were cherry trees treated the same as apple trees?
Couldn't tell you, but I wouldn't expect to find morels under them. But I never looked for a cherry orchard.
Yeah, I have found some under cherries.
Oh, wow. Good.
In a couple of different spots, Hunterdon County.
Wow.
And can I ask, if most of the time those apple trees have to be dead? Do you find more morels under dead apple trees or live ones?
To be abandoned orchards that are on their way out, but still alive, dwindling, very poor condition. They take a long time to die, and you should still see a few apple blossoms, and that would be when you would look for the morels, is when the little blossoms are out. But as the tree dies, And that's a place where you can find them year after year for quite a few years. I can think of a spot I used to go every year for 20 years and I would find some.
Yeah, I have my best orchard was one that I found in 1994. And I guess it was actually two orchards that are like adjacent, two different properties. And they're both small, you know, trees here and there, like 5-10 trees here and there in the woods. And when I first found this spot, I think there probably were about 40 apple trees scattered around. And now I'm down to 2 trees. So the trees are dying. And as the trees die, the fungus produces morels, the mushrooms. And as Sue just said, apple trees die very slowly. I've watched certain apple trees die over the course of like 10 years. And while they're dying, you'll get a few morels every year, maybe 4, maybe 5, maybe 10, maybe 20. When the tree is just about on its last leg and ready to die, is when I seem to find the most of them. And that's probably because the fungus knows the fungus has evolved in such a way that it knows it's losing its partner that produces its food. And so it just devotes all its energy to reproduction, puts out a lot of mushrooms. With the elms, it's a similar thing, but elms die very quickly. They're killed by Dutch elm disease. They die very quickly. So you tend to get not like 5 morels by a dead elm that's got the Marchella fungus on the roots. You know, you get like 50, you know, but all in 1 or 2 years.
The bark has to be, the tree has to be just recently dead. Usually when the tree is dead, but the bark is still on the tree for the elm trees. When the bark is completely gone off the elm tree, you're not going to find any morels at all.
Yeah, first year is best after it dies. They'll die very quickly. You'll see an elm tree that looks healthy, and a few months later, it's dead. I mean, I've seen that a few times. And then that first year, that's when it's best. And the second year, when the bark is still on the dead tree, like Susan just said, you know, sometimes I'll find a few more. But after that, it's usually no more.
All right, well, lots and lots of great information here, tips. I think we're about wrapped up here. I wanted to stop a minute early so I could give a couple quick announcements. I wanted to remind everyone this Saturday morning at 10 a.m. Eugenia Bone is doing a lecture, The Kitchen Mycologist, the delicious surprises, the utter flops, and the great tips and the ultimate lessons I learned from making our fantastic fungi community cookbook. That's her title. It's a long title, but she's a fabulous writer. I've read a few of her books. I've never heard her speak, but I've read a couple of her books on mushrooms, edible mushrooms, and she does some really neat stuff. So I expect that will be good. I know Elizabeth asked if it will be recorded. That I don't know. For all of these lectures that we've been doing, we have been recording them and sending the links out to them if the lecturer allows. So some of them, they let us, sometimes they don't. Sometimes people, I don't know, they want like rights to their material or whatever. So to answer your question, if we're allowed to record it, we definitely will, and I'll send the link out when I get a hold of it. Now next week, March 23rd for Taxonomy Tuesday, we're focusing on spring basidios. So, you know, all the non-ASCO mushroom that we would find in the springtime. And I was thinking we could maybe just go through and pick out like our five observations of things that we consider spring mushrooms, you know, that are not ASCOs, you know, all the guild and jellies and stuff like that, and kind of focus on that. And then after that, March 30th, we get back into the regular show and tell, showing off, because I expect some of us will start finding a little more stuff by then. All right. Is there any other announcements, or anybody else have anything else they want to add before we go?
Luke, I wanted to say that even though we're not doing an official foray this year for Morels, there's no reason. We usually go to the Princeton Institute Woods, and people can go there on their own and look around.
There you go. That's a good idea. So for anybody that's, you know, looking for a place to start out, to go to a place where you think you you know, where you have a good odds of finding a few morels. There you go. The Princeton Institute Woods is where the club usually goes. Usually we go there, what, the first weekend of May? That
Yeah,
seems to
always
be
the first Saturday in May.
the first Saturday of May. That usually be is a good time.
Sunday. Used to be. And we can all thank Joe Kaczynski for finding that place many, many, many years ago.
Oh, yeah.
Gone from now about 15, 20 years, but he's the one that found that spot because he's a bird watcher. And it's a great place for bird watching.
Awesome. All right. Well, then with that, I guess we will sign off. I hope everyone has a great week. Hopefully you can make the Saturday lecture. If not, I will see everyone next Tuesday. All right.
Will the Saturday lecture be recorded?
Thank you.
If
Bye.
we're allowed to record it, we will.
Perfect.
Sometimes the lecture allows us and sometimes they don't. So, all right. All
Thank
right.
you so much.
Bye-bye.
Have a good night, everyone.
All right.
Thank you. Nice seeing everyone again.
Thank you, everybody.